All right, good afternoon, everybody. I see that we have people coming in. We're going to go ahead and kick off our first seminar series. I want to welcome everyone to the Adventures in Field Research, our virtual seminar series that is sponsored by Lincoln Memorial University School of Mathematics and Sciences. What we're going to do is each week we've invited scientists to share stories about their research adventures, sort of giving us a peek behind the scenes of the scholarship of discovery. And so while you're listening to Dr. Galatowitsch's presentation today, if you have questions, you can feel free to use the chat box at the bottom if you're afraid that you will forget them. And at the end of his presentation, there will be an opportunity for you to unmute your mic and actually ask questions. So with that being said, we're joined by Dr. Mark Galatowitsch. He's Assistant Professor of Biology from Centre College, and he is our inaugural speaker today, and he's going to talk about There and Back Again, life history trade-offs and dispersal across dynamic freshwater environments. And with that, I'll turn it over to our speaker.
Okay, thank you very much, Dr. Rollins. Let's see, I'll get it up here. Okay, hopefully everybody can see that. Okay, so what I'm going to be talking about today is actually work I did for my PhD, and I attended University of Canterbury in New Zealand, and I was there for 5 years before I moved back to the United States to be a professor at Centre College. And what I'm going to do first is give you a bit of context in terms of the types of kind of broad concepts that I'm going to be covering, and talk more specifically about this, what's called a predator permanence gradient. It's really important in freshwater ecology. And then I'm going to focus a lot more on my specific New Zealand aquatic insect research. And I'll talk about the challenges and rewards along the way. And then I'll transition just a little bit at the end to talking about coming home and giving some tips I've kind of I've had for doing field research along the way. So first off, when we're talking about variable environments, it's really important to understand kind of how predictable or how long duration some disturbances might be. And so there's a lot of variability in the environment, and this variability in terms of disturbances can greatly influence the types of species we find in ecosystems, but also how able these organisms that live in those ecosystems are to evolve or adapt to those changes. And so while you might have some systems that are very predictable, and you might have daily disturbances in terms of tides, where you have organisms that might be above and below water multiple times during a day, versus other places, it's a bit more seasonal. And so like right now, we're in winter, and so we've got organisms that migrated away or have gone into hibernation, organisms that are reliant on seasonal monsoons. Versus other disturbances are less predictable and are more aseasonal, like droughts or fires. And then you also have kind of extremes where it's really unpredictable. They might happen every century or even millennia, like volcanoes or earthquakes. And I'm going to be talking more about these kind of aseasonal disturbances. But before I get to that, I'll talk a little bit about the seasonal disturbances too, and how they influence species distributions. Now, organisms have a variety of different adaptations to deal with disturbances, and some are really well adapted to this. And so we have some species like ponderosa pine that actually rely on disturbances like forest fires in order to be able to germinate. Or in other places, like in floodplain wetlands, in some places you have these seasonal dynamics where it'll flood for part of the year, and that gives fish access to those floodplains, and they can take advantage of some resources that they don't have in the main river channels. Now, this is a really important topic because we're now starting to see more frequent and less predictable disturbances globally, but also locally. And so understanding how adaptable organisms are to these volatile changes is really important. And so it's important that we study the resilience of these species to these disturbances and see the kinds of strategies that they might have to dealing with various different types of disturbances as a way to predict what might happen in the future in terms of the communities, but also specific species too. Now, my research focuses on freshwater ecosystems, and this is because they have really high biological diversity, and they're also some of the most productive ecosystems in the world. And there are quite a bit of changes that can happen in these ecosystems, from differences in terms of whether or not they're flooding or if they're going into a drought. You also have shifts in water chemistry or even changes in temperature, which can be major disturbances for the organisms that are living in those ecosystems. And what we can have are, you can have things that are kind of natural disasters, but you can also have just kind of normal ebbs and flows within these ecosystems. And so you can have these natural gradients of water permanence. And so I'm going to be talking about ponds for the most part, but we also have other systems like rivers, which can experience droughts and floods. In this picture in the top here, that's a braided river from New Zealand. Now this gives a good transition into what's called this predator permanence gradient that was proposed by Wellborn back in 2006. And I'm going to break this down and kind of build on it. And so this is going to be important because this is kind of a big central theme in my research to try to understand the kinds of stresses and how that's going to influence the life history strategies that these insects have. And so if we look at this bottom scale here in the bottom, we've got temporary to permanent, and then this kind of in between portion here, this is semi-permanent. And these are different types of habitats that have different hydrological regimes. And so a temporary pond might be one that only fills for weeks or months out of the year, but it will have a dry period. Semi-permanent might only dry every few years, but there'll be some years when it doesn't dry up at all. And then a permanent habitat that doesn't dry up at all only except for maybe really extreme cases might it dry up during a really bad drought. And so across this natural gradient of permanence, this habitat permanence, drying stress is obviously going to be the most important in a temporary pond. Versus in a permanent pond, it's not really going to be an issue for the organisms that live there. Now, this is also going to influence which top predators we find across this gradient of habitat permanence. And so in these temporary ponds, you might have just small invertebrates that are able to live in those habitats for short periods of time. Versus semi-permanent, we have some invertebrate predators like dragonflies that oftentimes need more than 1 year to grow there, and they might not be as well adapted to drying. And so that's oftentimes where they're going to be the top predator. In that drying, even though it only happens every few years, it can exclude some of the vertebrate predators like fish. And so in the permanent habitats, you'll have fish and dragonflies and a whole lot of other predators. And so that's why predation is going to be the most stressful aspect of living in a permanent habitat. Now, I was first exposed to this predator permanence gradient right after I finished up my undergraduate at Allegheny College. And I worked with Scott Wissinger, who's on the left there in that picture. And he was really interested in the invertebrates that lived in these alpine Colorado ponds and understanding how this predator permanence gradient influenced the species distribution of particular organisms like caddisflies, but also how other vertebrates, how some vertebrates like salamanders were also influenced across this gradient. So I'm just to the right of Scott there. And on the other far end there, you see someone whose name is Hamish Gregg, and he was from New Zealand, and he was doing part of his PhD in Colorado with Scott Wissinger. Now, Hamish's main PhD advisor, Angus McIntosh, was also there that summer, and this was my first exposure to kiwis, but also the research that they're doing down in New Zealand. Now, if we focus just on this Rocky Mountain Biological Laboratory research just a little bit, in these ponds, there's this complex of ponds across the gradient of drying and predator stress. So it's a bit hard to see in that bottom picture there, but there's a whole bunch of ponds that are kind of scattered there in the mountains, and these were created by retreating glaciers. And so we had some ponds that will only have water for up to a month or two, and then you had others that were semi-permanent, and you had these large invertebrate predators like these big predaceous diving beetle larvae. And then in more of the permanent ponds, that's when you had these salamanders, which are the top predators in these habitats because there were no fish. Now, these habitats, they're very predictable. You had the annual snowpack that that would envelop the mountains during the winter, and then a short summer period when these organisms would be basically rushing around and getting the resources that they needed. Now, across this predator permanence gradient, there were different species of caddisfly larvae. What Scott Wissinger had found, and I'd been helping out with in that research, is that there was these specialized life history strategies to live across this predator permanence gradient. And it's resulted in the species replacements where they're specialized to live in either a temporary or semi-permanent or permanent pond. And so if we look at that predator permanence gradient, I've replaced the predators here on the top, where you see these semi-permanent predators, these large diticid beetles, and they're really good at killing some of these other caddisflies that didn't have really good protective houses that they build around themselves. And so the ones that did the best was this caddisfly here in the middle that builds these really big blocky cases, and the mandibles of these beetles aren't able to stab through and eat them. Versus you have here on the other end, on the far left, in the temporary ponds, you had some caddisflies that were really poor adapted to dealing with other predators because they lived in predator-free environments, but were really well adapted at racing around and gathering resources and escaping. And then on the far end here in the permanent habitat, we have another caddisfly species that builds these long cases that were really well adapted to defend themselves against salamanders. And so I'm not going to be talking about my master's research. There was some time in the middle there, but when I was done with my master's, I was considering what I wanted to do for my PhD. And I recalled back to the work that I'd done in Colorado with Scott Wissinger, but also with Hamish Gregg and his advisor, Angus McIntosh. And I was really wondering about this predator permanence gradient, whether or not there were species that we consider generalist species that could actually somehow survive and actually exploit across this entire gradient of predator permanence. And then if that is the case, what kind of life histories might they have to balance the kind of trade-offs that they would have across this stressful gradient? Now, what's important to know about New Zealand is it's really remote. And so that is going to influence what kind of how many species are found in the island, these islands. But there's also this other factor too, where what they've found is that disturbances like the amount of rainfall that influences flooding and droughts is actually quite unpredictable. And so it's quite dynamic in New Zealand compared to somewhere like up in the high Rockies. And so what's interesting is that New Zealand temporary pond and river ecosystems have a high proportion of generalist species. And so it seemed like the perfect place to test whether or not there were these generalist species that could exist across this predator permanence gradient. And so I jetted off to New Zealand to start my PhD. And here's the connection to there and back again. It's kind of can't help but make some Lord of the Rings and Hobbit references along the way. So I was a PhD student who was enrolled at the University of Canterbury, and I joined the Freshwater Ecology Research Group, which had quite a few graduate students, and there was 2 PIs that led the lab. And in the bottom picture there, you can see us at the annual Freshwater Ecology meeting, where the annual dinner, there was always a dress-up component of it, so everybody dressed up in costumes there. But it was definitely a fun group. Now, New Zealand is a pretty amazing place. It's not a very large country. There's only about 5 million people that live there, but it has this kind of really amazing landscapes where you can go from down to the coasts up to the mountains, and you can move between those types of areas within just a couple of hours. And so it's really easy to get from the coast up into the mountains to where my research sites were quite quickly. And so my research focused on these pond complexes that were in the Southern Alps and looked a little bit like those that were in Colorado. Now, my research was actually building off of some works that Hamish Gregg had found during his PhD. He had since finished that he found. Now, example of one pond complexes in New Zealand, and we can see is there's a bunch of ponds that are scattered across this landscape, and they were left behind and they're created by glaciers as they retreated. And so some of these had much more kind of porous ground, and others, it was a lot more permanent. And what he also found using water loggers is he measured kind of how quickly they filled and how quickly they dried. And across 2005 to 2007, we can see in the green there, this is the number of ponds that are filled, and then the weekly rainfall is in the black. And so we can see from year to year, there's actually quite a bit of variation in terms of the rainfall, but also how many ponds were filled. And he made this connection between kind of this really dynamic kind of hydrology across ponds, and he found that if you looked at the freshwater invertebrate communities, that there was this higher number of generalists than what he was seeing in places like Colorado. Now, this is a Venn diagram where on the outside there, these are organisms that are going to be specialists to either the temporary, semi-permanent, or permanent habitats. And then you have overlaps where they're able to survive across those different types of ponds. And what's kind of really interesting was this complete overlap. So we had 27 species that were generalists across this whole gradient. And so this is what I was really focusing on, is trying to understand what are some of the life histories that they would need to be able to live in a place that has fish, but also that same species that could survive in an area that might only have water for maybe a month or two out of the year. And so I had come up with a variety of different for what I might expect for organisms that were basically having to deal with this kind of really contrasting types of stresses. So I expected that individuals of that species that were living in a temporary pond, that they would probably grow fast and or have an ability to tolerate drying periods if they're found in a temporary pond. Versus if you have the same species that was living in a permanent pond, that they probably for predator avoidance. So they might move less, which could slow them down in terms of how quickly they could gather resources, but they could be slowing themselves down in order not to be noticed by visual predators like fish. But it also seemed really important that these organisms might be exchanging individuals across this whole gradient. And so this is what's allowing to be a generalist in habitats that are temporary or replace individuals that might have been lost due to heavy predation by fish. I focused on two different species, and so I was more at the community level rather than, population level rather than community level that Hamish had. I focused on a species of boatman and a species of damselfly. Now, both of these species are widespread throughout New Zealand and are common across predator permanence gradient. Now, it works in the same pond complex that Hamish did, but I also added a couple more because it was important to me to be able to see kind of how broad these kinds of strategies might be. It might be that you just have species that's really specialized in one pond complex and it might not actually apply to other. And so it was important to see kind of how applicable it was across a large range of the Southern Alps. And so we can see here is we've got the city of Christchurch where the University of Canterbury is, and then these three different spots where I would go to collect my invertebrates. And so my main methods were a multi-year field study of the populations of those two species across that pond permanence gradient. I also followed this up by doing Mesocosm experiments to really kind of empirically test to see if there was this potential flexibility for predator avoidance, how changeable their growth rate might be, and whether or not they had an ability to tolerate drying. And I also wanted to see how likely they were to disperse away from some of their habitats. And so I installed some artificial ponds, but also did some population genetics to test for potential dispersal patterns among the different ponds, but also even between some of these pond complexes too. So I had a plan, I was all ready to go, and I had arrived in August of 2010. But right at the beginning of September, there was a massive earthquake that had hit Christchurch, and it was a 7.1 earthquake, and had done little bit in terms of how well buildings held up. Nobody had been killed. There were some people that had been hurt by the earthquake, but it did cause a big kind of pause in terms of the research. In terms of they shut down the university. Basically, we all had to be remote for a couple of months while they try to figure out how safe it was to occupy the buildings. And then there was a subsequent aftershock, which was really strong, 6.2, which it wasn't as strong, but it's a lot more shallow and caused a lot more damage. And about 200 people died from that. And by the end of my time in New Zealand, I'd experienced about 10,000 4-point or above earthquakes from living in Christchurch that have been triggered from this massive initial one in 2010. So it was a pretty dynamic time to be in Christchurch. But there were some delays, but fortunately, I wasn't personally affected, and the people I was working with hadn't been either. Was quite disrupted, and so it wasn't just physical damage to buildings, but you also had this weird thing that I didn't know about before this, which is called liquefaction, which causes basically silt to bubble up from underground and basically makes everything almost like concrete, because the city is built on alluvial plain on this kind of buried sand. But eventually I was able to really kick off my research. And so I was visiting these 3 pond complexes, and I sampled 34 lakes and ponds every 2 months for 3 years to really monitor these population dynamics over time. Now, because I was going up into remote mountains, I also had to take 4-wheel drive training and also learn how to drive stick on the left side of the road. And so that was stressful at first, even to get around roundabouts in the city. And then once I was out in the mountains, it was actually quite a bit easier. But sometimes some of these mountains, you'd have landslides that would come down even when I was at some research sites, and so I had to be prepared to get creative to get back in and out of the research sites. I did not flip this van at the bottom, but just highlighted that it could be a bit dangerous driving on some of these mountain roads. Now, where I was in the Southern Alps, a lot of my work focused at the University of Canterbury's Cass Field Station, which is there at the top. And there's this old historical building where I spent a lot of my time there. And I did my Mesocosm experiments at this field station. Now, I also worked out of some Department of Conservation huts. And so some of them were a bit bigger, had some kitchens, while others were essentially just a couple of bunk beds, and I had to bring camp stoves. And in each of these places, I would go up there for a week or 2 or even months at a time. Now, as I mentioned, New Zealand is quite unpredictable, and this could be quite challenging. And so you have this really kind of crazy weather. And so in this picture there, I'm working in one of the remote sites I had. And some of this weather, it would be during the summer, you'd see some weird clouds moving in, and it would start to rain, but then it would start to snow. And then that cloud would pass and it'd go back to being about 80-degree weather. And then you see another cloud move in, and you'd have like basically 3 or 4 seasons over the course of just a few minutes, and so you had to be prepared for whatever was going to happen. And so definitely had to be prepared at all times, had to have coats, but also be ready to take it all off and have t-shirt and shorts. Now, because we had this really kind of wild weather patterns, you also had lots of differences in terms of precipitation. And so the water levels could fluctuate quite a bit throughout the year. So this is a picture of one temporary pond where it could go several meters deep to completely dried up within a few months. And this was actually a site that I was able to collect both of my organisms from 1 year, but then the subsequent 2 years, I can only collect the water boatmen and not the damselflies. So sometimes I would show up and the ponds would be dry or almost dry, and I would have to sift through mud and try to find my invertebrates. And so you can see my legs there. Sometimes I get quite muddy, up to my knees or higher in mud, trying to sample these places. And even the permanent ponds, you can have really big fluctuations in terms of the water level. And invertebrates like to live on this vegetation that grew on the lake bottoms. And what would happen is the vegetation wouldn't move very much. And so the invertebrates would stay in those places, and I would basically just try to get to that vegetation. So sometimes it was quite easy. The water levels would only be knee or waist deep. But what I learned is during the winter, water levels would rise in these lakes quite a bit more. And so I couldn't use my hip boot waders or chest waders anymore. I actually found out I would have to completely cover myself in neoprene. And so I started using free dive suits, and I would actually go out and bob around up to my shoulders to try to get to the insects, because I wanted to get this kind of this time series with these invertebrates to follow them and see how quickly that their body sizes were changing over time. And sometimes I even had to bust through ice to try to get access to the invertebrates that were underneath there. So actually, in this picture here, I broke holes through the ice and then I actually jumped in there. I've got the full body neoprene on so I could try to collect my invertebrates. And so it was sometimes pretty wild, and I didn't know what I was going to expect when I went up to these mountain ponds. Now, what I was doing was I was actually collecting about 100 nymphs per site every 2 months. And so I was there throughout the year. And then what I would do is I'd collect them and measure them as a way to kind of follow what their cohorts, how their cohorts might change over time in terms of their body size. And so I'd graph these out and basically try to figure out when you might see different cohorts through time. And then basically construct and figure out kind of how quickly their growth rates were. And so I did this for 32 ponds across those 3 different years I was sampling. Now, I'm not going to go into a lot of the details in terms of what kind of patterns I found, but just kind of to cut to the chase, for the damselflies, they could complete about 1 generation per year in the temporary ponds. But if there was a pond that had too long of dry periods, they would be completely excluded from that pond unless it was a more wet year and they could eke out a generation. Versus the water boatmen, they could generally get about 1 to 2 generations a year because they had a much shorter development time. Now, in permanent ponds, there were no time constraints, so these were not drying up. And the damselflies would actually take 2 years to complete 1 generation. So it's a big difference in terms of how quickly they're growing. Versus the water boatmen, they would still basically kind of crank along and they could get through about 2 generations a year. And so broadly what I found, especially for the damselflies, is you see this accelerated growth in the temporary ponds, but also some evidence that they might be dry tolerant, versus in the permanent ponds, they definitely had evidence of much slower growth. Now, I also did mesocosm experiments, and so this is back at the Cass Field Station where I'd set up 40 different tanks, and then what I did is I had different treatments in terms of different types of predators and exposed my invertebrates to those predators to see how well they might be adapted to that. And I would take organisms I collected from temporary ponds and also ones from permanent ponds to see if they had different strategies in terms of avoiding predators and how vulnerable they were to those different predators. I also did behavioral experiments too to actually watch and see how much they're actually moving around if they felt exposed to these predators. And what I found was those that were found in temporary ponds seemed to be quite susceptible to fish predators, and they seemed to be pretty naive. So if they were sourced from a temporary pond, they seemed to be pretty naive to predators like fish. Versus those I sourced from permanent ponds, the damselflies seemed to have this adaptive predator avoidance. They moved a lot less, and so that could give them an advantage to not being spotted by fish predators. Now, in contrast, the water boatmen, they didn't seem to change their behaviors at all. So they didn't seem to have much of a variable kind of strategy for living across this gradient. I also did some laboratory experiments where I created these very fancy, kind of more smaller mesocosms where I basically carried some pond sediment back to the lab, put them in these containers, and then dried them up over a period of time. And I basically measured to see whether or not they were able to survive periods of drying. And what I found was that the damselflies seemed to be adapted to drying. About 80 to 90% of them were able to survive at least 8 days without water. And this is important because there was a time when these ponds would dry up, you might get a little bit of rain, they would have water for a bit of time, and then it might dry up again. And so even having an ability to survive 8 days or a bit longer without any water really kind of gave them an edge in those temporary ponds. The water boatmen, on the other hand, they couldn't handle the drying at all. And so if they didn't have wings and they weren't adults, so they couldn't fly away, you had 100% drying mortality. So the two strategies that I found for these generalist species seem to be quite different. And so the damselflies, overall, they have a longer developmental period compared to the water boatmen. And so they have longer exposure to both abiotic and biotic stresses, even in the temperate ponds. And so they seem to have greater developmental and behavioral flexibility, and they also had some evidence of drying tolerance. In contrast, the water boatmen, they were actually able to exploit more temporary habitats. They could quickly get into these habitats, quickly grow, and try to get out as flying adults and move on to maybe a more permanent habitat, and they would be able to escape that way. But if there was a habitat, like a permanent habitat that didn't have good vegetation or nice kind of slopes where they could hide from fish, they could actually be excluded from more of the permanent habitats due to their less flexible behaviors to avoid predators. And I also see how this kind of adult dispersal between the different pond types might be important to sustain the distributions of these species. Now, I anecdotally had observed this happening, especially with the damselflies, where I could actually follow eating damselflies, and the male actually holds onto the female, and then the female oviposits or lays her eggs on vegetation. And so these are actually ponds that weren't too far away from each other, and I could actually follow the damselflies and see them moving back and forth between these ponds. And so that seemed to suggest you could even have an individual female that wanted to lay her eggs in a temporary pond, but also in a more permanent pond too. Now to test this, I built artificial ponds to see how far they might move between a permanent habitat and something that maybe seemed a bit more novel. And so I set up these artificial ponds or these cattle tanks, filled them up with water, and set these up 100, 500, and 1,000 meters away from a permanent lake. And there weren't any temporary ponds that were located near them. But what I found out was this was a real big pain. I mean, I had to haul lots and lots of water up to 1 kilometer away from these lakes. So I was driving across the land and actually blew a few tires along the way because there's some really nasty spiny vegetation that would puncture the tires. And sometimes I would arrive after driving a very bumpy ground. I would get there and half the water would be gone. So I'd have to go back to the lake, fill up more barrels and drive them up to these. And because you didn't always have rain to replace that water I put in them, they would dry up unless I went up there and constantly refilled them. And another issue I came across was some of these areas where I put these tanks was also private land where there were cattle. And what I found out is cattle are really curious and they really like to drink water. And so I would sometimes come up and I'd find that the cattle had basically just pushed it over, played with the tank, or just drank all the water completely. And so this experiment actually was pretty much a bust. It was just too tough to keep water in there and too tough to try to keep cattle away from them. But I also did population genetics to see kind of more longer term patterns in terms of the relatedness of the damselfly individuals across these different pond complexes, but even between them too. I'm not going to get into a lot of that detail of this. Basically used a lot of those individual damselflies that I collected from my field survey to do these microsatellite genetics. And I had done this across the 3 pond complexes in the South Island, but also included one on the North Island. And what I found was there really wasn't variation between the pond complexes between them or even within them on the South Island. There was a difference from the North Island though. So it seems that the ocean actually is a barrier to these damselflies, but the mountains might not actually be too much of a barrier even for an insect it's not very big, but they might be strong enough flyers and have just enough exchange of individuals that we're actually getting quite a bit of mixing going on. And so it doesn't seem like there's much habitat isolation, particularly for these damselflies, and extensive gene flow within the pond complexes. And so this could be really important for sustaining these populations across these pond complexes. Now, also when I was in New Zealand and I've been collecting lots of invertebrates, there was another research group that was working up a couple of floors above me, and they're actually focused on viruses, and they were finding all kinds of cool new viruses, and they're actually going to some pretty extreme places to find some of these viruses. And I'd heard a talk in one of our departmental seminars, and they talked about how they're going to places like Florida, to Tonga, to collect dragonflies, to test for novel novel viruses. And I approached them. I said, you know, I get all kinds of dragonflies all the time when I'm collecting. And they were quite intrigued. So they said, well, bring in some next time. But they didn't expect me to just bring in about 50 dragonflies when they'd always struggled to even get just a few. And so they were pretty shocked that I was able to collect so many of them. And for me, they were just bycatch for a lot of what I was doing. So it's really easy for me to collect them. And so we actually found some novel viruses. There are these circular DNA viruses in these dragonflies. And then we subsequently did another project where we actually looked at a whole variety of different organisms living in these New Zealand lakes. We did damselflies, water and sediment, and other organisms too, to see if there were these kind of shared viruses across these different organisms, and also from the sediment and water. And so that pretty much kind of very quickly sums up my research that I'd done for my PhD in New Zealand. And I took that knowledge that I had from New Zealand, and I had gotten a job at Center College in Kentucky, where I'm still at now, and I'm an assistant professor there now. And I'd carried a lot of that baggage back there with me, and kind of taken a lot of that knowledge that I had from Colorado, but particularly from New Zealand. And now I'm applying it to some of the temporary streams that we have here in Kentucky. And so now I'm looking at temporary streams that not only have to deal with drying for part of the year, but you also have flooding and fast flowing water, which is actually even more challenging for some of the invertebrates. And so now I'm trying to find, okay, our invertebrates have here in Kentucky that might be similar to those that we see in New Zealand. Okay, so I just wanted to finish up by talking about some kind of tips for maybe for your field research. And so what I learned through my time is it's really important to build on the foundations that others have laid. And so my work built off of these ideas that had come out of Wellborn, Scott Wissinger, and Hamish Gregg. It's also important to know that you're part of a community. And so it's also important to rely on your networks. And so Scott Wissinger had been one of my undergraduate advisors, but he was a really good kind of avenue for later where I went for my master's, and particularly where I went for my PhD. Also, it's really important to treat field assistants with respect. And so it's important to feed them well and pay them if it's possible. And I definitely saw this with lots of different other field crews that were out there, where sometimes you'd hear people kind of grumbling how they were just eating peanut butter sandwiches every day for lunch and dinner, and they were doing it as a volunteer. You could see that they'd really kind of get frustrated with it, and it probably was influencing the quality of the data that was being collected. And so I put a premium on really good food, even if it was on a camp stove out in the middle of the mountains. And also it's important to just roll with the challenges and also see what might be open for new opportunities. And so I definitely wouldn't have thought of doing any research with viruses, but it just happened to be that I was talking with other people in our community at University of Canterbury, and we found out this kind of other cool avenue we could go into. And then obviously rolling with challenges is really important for working in these really dynamic kinds of ecosystems. And then along the way, you build up lots of new experiences, and they can be added to your toolkit, and you can take them on to your next steps. Now this research, there's lots of pictures of just me, a lot of people that were involved along the way. And this was through field help that I dragged out to go sample in the snow, or when it was really hot out up in the mountains. But there was staff that was at University of Canterbury that were really important for me to do my genetics research and help support the field research too. And so that's what I've got for my presentation. So any questions? Sorry, I wasn't monitoring to see if there was any comments that were coming in.
Mark, I've got one for you. This is Shay Tuberty from App State. I really enjoyed your seminar.
Thank you.
A lot of familiar work that you've done, and some new things I learned as well.
Nice.
So just a technical question about water boatmen, adults. You mentioned wings. I immediately jumped on the internet and was like, what are you talking about? Do they fly, or are they able to use their wings in some way? I wasn't aware of that.
Yeah, so water boatmen, they're not very big, but they do have wings and they can move around. And so there were times when I would actually be out in the ponds, and there was one situation in particular that was kind of bizarre, where it was a pond that was drying up and it seemed to be drying up pretty fast. And all of a sudden I saw like basically the water was bubbling up. And what I saw was a lot of these water boatmen starting to shoot up into the air. And so they were flying away. I'm not sure what kind of triggered them for that suddenly do that, but this is why in some of my experiments, I definitely had to make sure I didn't use adults with because they had wings, because I was worried they might fly away. Now, in New Zealand, those ones, they definitely have wings and they can fly. In other places, you actually have, there are some morphs that actually have reduced wing muscles. And so they might have really rudimentary wings or wings that are maybe a bit more protective, but they actually can't fly very well with them. But the ones in New Zealand had fully functional wings.
Thanks. I was very unaware of that.
Yeah.
A little embarrassed not to know it, actually.
No worries. Okay, so what is the benefit of slower development in damselflies when they are in permanent ponds? So the thought might be is you've got predators like visual predators like fish that are basically looking around and trying to notice movement in order to find their food. And what might be going on is if you're racing around, and you could actually be drawing a lot of attention to yourself. And so if you move a lot slower, or if you're a bit camouflaged, you might not notice, your predator might not notice you. And so by not kind of moving slower, not being noticed, it could also reduce your foraging rate too. And so I didn't actually directly measure foraging rate. I did measure things like how much they were moving over periods of time in some behavioral experiments I did. And it seemed that the on damselflies definitely had these behaviors where they moved a lot slower, and particularly when they're exposed to fish. And so if they're moving slower, maybe they're foraging less, and then that might also reduce their ability to get the resources that they need to reach particular developmental stages. And so that's what could be slowing them down, versus in a temporary pond, they might just have that kind of immediate stress of drying up. And with fewer predators around, they might race around and try to gather up as much food resources as they can. And that also might allow them to develop faster.
Can I ask a quick question? This is Joe De Silva.
Yeah.
Hey, Mark, I thought that was very interesting because you talked about challenges and you talked about uncertainties that confront all of us that go out and work in the field. And you covered a lot of miles, didn't you, going up to New Zealand?
I did. It was lots of driving, also a lot of hiking. Sometimes I could only drive to a certain point and then had to basically hike in with all my gear and carry out all my samples out.
And then seasons as well to contend with.
Yeah. So that was quite challenging, particularly during the winter, to get up to some of these sites.
So the question that I had for you is about generation time. You said that generation time in temporary ponds was quick, shorter than the ones in permanent ponds.
Mm-hmm.
So when you say generation time, are you talking about life cycles or what are you talking about?
Yeah, and so I was talking about basically going from an egg stage all the way to an adult, that it was a much shorter period of time to make all those developmental stages. And it was much shorter in a temporary pond compared to a permanent one, based on the kinds of cohorts that I was trying to follow through time.
So what is the generation time for your damselfly that you're talking about?
So it seemed like they had to have at least 1 year. And so they needed to be able to go from that egg stage to an adult. And for some of them, if they encountered a dry period, some of them were able to survive that dry period, versus others, if it remained dry for too long, they would die and they wouldn't be able to complete their life cycle. Versus in a more permanent habitat, it seemed like they were actually taking longer than a year to complete their life cycle.
So the next question that I had was, did they strategize? The population, let's say in a temporary pond, would they go over to a permanent pond if they could, if they would fly over?
So that's, yeah, that would have been really kind of difficult to do in terms of it's hard to know and be able to follow individuals. And so I was trying to see if there were patterns in terms of the microsatellite population genetics data to see if there was isolation in terms of if there wasn't much mixing going on between the ponds, we would have seen maybe more distinct population genetics in a temporary pond versus a permanent versus temporary pond, and maybe they weren't mixing as much, but it seemed like that they were moving between them. Now, how much an individual female was actually laying eggs across those and actually doing bed hedging with her offspring, it's hard to really know. I potentially could have sat out there with in some lawn chairs with some marked damselflies on their wings. People have done that before with dragonflies, and you actually just sit out there with binoculars. You just basically watch and see where they're going and what they're doing with their behavior. But that would have been tough in terms of seeing how much they're moving between these ponds versus just sitting at one pond and monitoring their behavior that way. And so it would have been tough to kind of figure out what each individual female might be choosing. But I'm particularly interested to know if there is a way to do that. These insects were too small to actually put little radio transmitters on, but that would be cool to see if that's possible.
Thank you.
Yeah. So actually, so there's a question, there's a comment here. So I'm always removing boatmen from my pool and they return the next day. So one experiment that I didn't actually mention was something I did based off of the PhD student there who's kind of huddled up there in the snow. She was doing research at the Cassfield Station 2, and she was doing subsidy experiments, and she had actually built these really big cattle tank habitats, and she was monitoring the communities over time in those. And what we actually tried to do to see how much insects might move even between her tanks, is we actually went in and then gathered back swimmers, and then we used Sharpies and actually drew on them and put them in these different tanks to see if they would actually move the tank that fits them. And what we found was that didn't work particularly well because they're really fastidious and actually found ways of rubbing off the markers off their wings. So that was another kind of a bust experiment, but that was one way we thought maybe of trying to track individuals from one place to another, but it didn't work out for us.
Well, I have a question. Very much enjoyed your presentation and happy that you have provided this information. I'm curious, your work that you talked about occurred in New Zealand. Did you encounter any sort of cultural challenges working in that country?
I really didn't. I mean, the only cultural challenges was just trying to figure out how to understand Kiwi English. And so, yeah, it was just trying to particular words that I didn't quite understand. Took a bit to figure out. So like plastic containers called bottles. It took me a bit to figure out what that was. It was really pretty basic, and they spoke English, so that made it really easy for that. And it was much easier than other experiences I've had in the past. So right after I left Colorado, I actually did Peace Corps for 2 years in Nicaragua. And so I taught elementary schools in rural Nicaragua for 2 years. And that was a lot more of a challenging cultural shift. New Zealand, it was a lot easier to move there. It was really easy to live there, and didn't have any cultural challenges really. And actually, I'm a bit jealous now because they've been so good at keeping the COVID cases down that I see them with pictures where they're all kind of at picnics, going to rugby games, lots of people interacting without masks on, and it's a really great place to visit, and they have a really kind of great community, and everybody seems to be working together, particularly with trying to keep COVID cases down.
I'd just add to that question, Adam.
Yeah, go ahead.
In the North Island, the Maori are much more prominent, I think. And especially where their national parks are largely owned by Maori organizations. They're indigenous people, they're First People. And many of the places you visit still have the Maori names. Cities still have the names. I noticed that's still happening in the South Island, Mark. I take classes there, and we're not doing research per se. We're teaching field work. So how to do a transect, how do you monitor and measure biodiversity? And in doing that, you allow the students to kind of enjoy all they have to offer as far as their habitat types and the diversity. And I just wanted to say that, you know, it's no joke about the dangers of doing field work. Even if you don't think there should be any danger where you go, you never know where there's going to be a landslide. And both in the North and the South Island last year when I was there, we had to deal with major road blockage. And we were lucky that there was already big machinery fixing the roads so we could get through. But we happened to be on the South Island going into Milford Sound. We were waiting at the stoplight at the tunnel you're aware of, I'm sure. And, you know, major rain. We got 1 meter of rain that weekend, 3 feet of rain in 2 days. And there was a waterfall every 100 feet coming off the side of the mountains, just amazing. But the clouds were just above the height of the bus we were in. And so we're standing on the side of the road, taking pictures of the waterfalls. And it sounds like a 747 passing over. And it's literally the top of the mountain falling down the mountain, bouncing off the rocks, you know, so just tumbling down. And eventually you could start seeing Volkswagen bug size rocks bouncing down the mountain right at us. We took about a 10-foot leap backwards and had rubble rolling to our feet. So I think some of the kids wet their pants, others were jumping in joy because it was so exciting to almost die. But, you know, in the end, when I got my student evaluations, they kind of were upset about it, that I would take them someplace so dangerous. And I was like, well, how do I know? I'm on a major highway, you know, a two-lane road to nowhere. But still, it's no joke. When you're traveling in these places, going to remote field stations, you got to know that there's a chance you might not come back.
Yeah, and that was actually something that University of Canterbury and the New Zealand in general takes really seriously. And so whenever I'd go up to these field stations, I'd have to fill out really detailed forms in terms of where I was going to be, which route I was going to take, and how I was going to get out. And every single day I had been taught. Now that's challenging in some of these places because I was in remote mountain areas with no cell signal. And so I had a satellite beacon that I would send messages out every single day. And if I didn't send out a message by a set time, that would trigger people following up and even sending out search parties. And so also had an emergency beacon. And so I know that there was, when I was there, at least one group had pulled the emergency beacon, and they actually, people had to go and check on them. And there was actually helicopters that were deployed to find them, and they were safe. It was actually just an accident, but they take it really seriously. And so from those earthquakes, when I was there too, that actually closed down one of the field stations they had on the coast. It's now permanently closed. And yeah, so fieldwork in New Zealand can be dangerous, and even historically, what was called the New Zealand death was people who had drowned from some of the rivers and how they quickly would flood. And so it could be challenging to do field work there, but there was quite a bit of a safety network, and healthcare was really good. I actually had an accident when I was just doing some vacation hiking. I had a serious accident with my foot, and I had to be hospitalized and had some surgery on it, and ended up only costing me not even $200, even though I'd spent several days in the hospital and had major surgery. And so the healthcare system was really good, and I kind of miss it sometimes, being back here in the US. So.
Hey, thanks for that talk. That was amazing.
Yeah, thank you.
Makes me miss field work, but so I studied abroad at Lincoln, and I know I did some wildlife conservation, so we did some field work at Kaikoura, which was very brief but amazing. So how did you land on University of Canterbury for the students that might be interested in getting some research abroad or kind of broadening their horizons?
Yeah, so how I found out about University of Canterbury doing research in New Zealand was from that time that I was working in the Rocky Mountain Biological Lab, and I'd met Hamish Gregg and Angus McIntosh, and Angus was eventually my PhD advisor. So that's initially how it got on my radar. I think even if I hadn't met them and I'd later found out about potentially doing PhD research or doing research in general in New Zealand, I definitely would have been very eager to do that too. And so universities in New Zealand, if you go there as a PhD student, you actually pay the same rate as you would if you're attending as a domestic student. Master's students, they have to pay a bit more if you're an international student. Now, how I was supported while I was there, had from the University of Canterbury, and I actually stumbled across another fund when I was trying to fill out my my visa to go live there. I stumbled across this one really lucrative scholarship that they had that was actually funded by the federal government. So that was New Zealand Educated. And I applied for that one too, and actually ended up getting 2 scholarships. So I actually had to give some money back because they couldn't let you get rich off of being a student. But that was a great way to support myself while I was there. Now, another thing that New Zealand has, so if you're not thinking about doing research as a graduate student there, is they also have really good programs for people that are, I believe it's under the age of 25, where you can actually go and work there and you can get a short-term work visa. And so there's programs that you can do for that. So you don't necessarily have to be in a university, but there's ways of actually getting there and getting visas to live in New Zealand, to do research, or even just to live and work there in other fields too.
All right. Well, I think we're down to the wire here with our time, so we'll
Okay.
thank Dr. Galatowitsch one more time for agreeing to present. Very, very enjoyable. And of course, you can reach out to him if you have additional questions. I'm sure he'd be happy to talk in more detail.
Yeah, before it gets closed here, I'll put my email address in the chat. Okay, and I also want to thank Dr. Rollins for organizing this really cool seminar series. I look forward to attending some of the others in the future. So thank you very much.
Thank you, everybody, for attending. I do want to thank Lainey and John, who are instrumental in putting this together. If it were not for the 2 of them, we would not be able to enjoy each other's company and learn the information that we're learning. Additionally, I would like to put in a plug and a plea for next week. We'll meet weekly. I'd like for you to invite a friend. I'd like you to register yourself and encourage your friends and colleagues to register for the next talk as well. Go ahead and follow us on Facebook or Instagram. You can follow Lincoln Memorial University or the School of Mathematics and Sciences. And we'll leave you with this. Next Wednesday at the same time, we have Dr. Amy Boyd from Warren Wilson College, who's going to give a presentation titled Death Watch in the Desert and Other Adventures. So thank you all. I hope to see you next week. Have a great evening.
Thank you.
Bye-bye. Thank you.
Thanks again.
Yeah, I think it went well. Thank
Yeah.
you.
Yeah.
See you all next week.
See ya.